Hello everybody and welcome back to your online lecture. This chapter, chapter 17, is your final chapter for this course, so congratulations, you made it. In this chapter, we are going to be looking at gene expression. So this is it. You've learned about cells, you've learned about the organelles, you've learned about the macromolecules and how they're important in their various functions and locations within the cell. We learned about energy, learned about mitosis and meiosis, we learned about the mitochondria and cellular respiration, chloroplasts and photosynthesis. And finally, we're at the point where we can put all of this together and learn about how all of these genes are actually expressed so that our cells can do all of this, because all of those proteins, everything that's involved with what we just talked about is encoded in the DNA. So, gene expression. We're going to go through transcription. We're going to learn about the genetic code. We're going to learn about mRNA modifications. And then we're going to learn about translation, where we're actually building our polypeptide, and we'll go over a little bit about mutations. For this chapter, students will describe or model the regulation and expression of genetic information, particularly describe or model the transcription process contributing to gene expression in eukaryotes, describe or model the translation process contributing to gene expression in eukaryotes, and describe, model, or map DNA mutation and its consequences in gene expression in eukaryotes. So as we know now, the DNA is probably the most important molecule in the body because without it, no other molecules would exist. The DNA is important because it is what contains our heritable information. It is our genetic material. So it contains specific sequences of nucleotides. Remember, the nucleotides of DNA are A, T, C, and G. So we can have different sequences of A, T, C, and G, which are coding. They're going to code mRNA, which make us proteins. We know that DNA is inherited by organisms, right? It's passed down from the parent organism. And those specific traits that the DNA is encoded dictate the synthesis of proteins. Proteins are the links between genotype, what is encoded in the gene, and the phenotype, what we see. And we know that proteins have an abundance of jobs within our cells. If you need to look back at chapter 5 and see some of those jobs for proteins, look back now. And we've already talked about gene expression. Gene expression is the term used to describe reading the DNA, which is a gene, and turning it into a protein, which is our cell expressing that gene. We have thousands of genes in our DNA, and most of them are not being expressed at one time. They'll be silenced or turned off. We have to particularly purposefully express a gene. So this is gene expression. Gene expression is two distinct stages: transcription and then translation. And we now know that one gene, one protein is how it works. So for one segment, one sequence of the DNA, which is a gene, it creates one protein, or one gene, one polypeptide. Remember, the polypeptide is just the polymer of amino acids. So it's the linear chain of amino acids. And we have to fold that properly. Again, if you need a refresher, go look at chapter 5. We need to fold that polypeptide properly in order for it to be a functioning protein. But it's one gene, one sequence per protein or polypeptide. So let's just cover the basics, some of those words that I was just using transcription, translation, mRNA. So this schematic that you see down here, DNA goes to RNA, goes to protein. This is called central dogma, the central dogma of molecular science. And it's the concept that cells are governed by a cellular chain of command in that particular order. DNA leads to the creation of RNA, which leads to the creation of protein. So the first step, transcription, is this arrow right here. Transcription is reading the DNA and from it creating RNA. So here's transcription, the synthesis of RNA using information in DNA. We've already covered some types of RNA, or you probably have heard of them, but just note that we're going to be talking about 3 particular types of RNA in this lecture, and you have to know each one. So particularly, we're making mRNA from the DNA. M stands for messenger because it contains the message of the genetics within the DNA. So DNA gets transcribed into mRNA. The next arrow over here, this is the process of translation. Translation is the synthesis of a polypeptide, which is a linear chain of amino acids, using information carried by the mRNA, our messenger RNA. Right? So the mRNA is going to be translated into a polypeptide and folded into a protein. The part of the cell that is responsible for translation is called the ribosome. And we did cover ribosomes a little bit in chapter 6. We'll look at them again in this chapter. But just note that the ribosomes are what are actually translating the message carried in the mRNA into the polypeptide, the protein. I'll also probably use this term transcript. So we're going to revisit this again as we go through, but this arrow here again is transcription. When we transcribe DNA, we create a transcript, and it's called a primary transcript because we have to take that initial primary transcript and we're going to have to process it and we're going to modify it a little bit more before it becomes the true mRNA that goes to the ribosome to be translated. But we'll see that. Now let's look at the difference between a prokaryotic gene expression and eukaryotic gene expression very quickly. Remember, prokaryotic, this bacterial cell over here, they do not have their organelles. So all of this can occur more simultaneously than in a eukaryotic cell. You'll see here that transcription is occurring in the nucleus. So we're reading the DNA and we're creating our primary transcript, our pre-mRNA. We modify it into the final mRNA. That transcript has to leave the nucleus before it can find a ribosome and become translated. It's a little bit different if you don't have a nucleus, as this bacterial cell here. So it says in prokaryotic cells, translation of mRNA can begin before transcription is finished. So these two processes can occur at the same time. This is not the case for eukaryotic cells because of their separation of those two stages. Okay, so we are going to walk through transcription and translation, as in which enzymes and transcription factors and these different proteins that are involved with these steps. But first, we're going to look at the DNA, the mRNA, and the protein, and look how they're all related. How is it that the DNA is coding to create a protein? So to look at this, we have to revisit the nucleotides that we learned about in chapter 5. So we know that the DNA is comprised of A, T, C, and G, and we see this over here. We have a double-stranded DNA molecule. We see, just as a refresher, that they're anti-parallel. This is 3' to 5', and then we have 5' to 3'. So they're anti-parallel. There is complementary base pairing because A is always bound to T and C is always bound to G. Good. And we see that one of these DNA strands is called the template strand or the non-coding strand. It has a couple of different names up here, antisense. And the other strand is called the coding. So there's the template, which is non-coding, and then we have the coding. So this is important because the one that is red, because only one of them is red to make an mRNA, the one that's red will be the template strand. And we're going to see that the coding strand is very similar to the RNA transcript that's made by reading the template strand. Because again, we're going to be utilizing complementary base pairing. So, the DNA template strand is used to create this mRNA strand. Notice that the mRNA strand that's created is in complementary base pairs to the template strand, with the exception of DNA has A, C, T, and G, whereas RNA, the nucleotides are A, C, U, and G. So DNA has T and RNA has U. So here, the A is going to be complementary base paired to a U to make mRNA. C is still paired with G, C with G. Here's A with U, A with U, A with U, C, G, C, G, C, G, A with U, C with G, T with A. Okay. So this is the transcript. This was the process of transcription. Now we have this transcript. So the way that these transcripts are written and read are in groups of 3. And those groups of 3 base pairs is called a triplet code, and it makes up a codon. So we see here one codon. This codon is UGG. Next to it is the second codon, UUU. Then the third codon, GGC, and then the fourth codon, UCA. So these codons kind of act like words, they'll say. These codons code for a specific amino acid. And remember, there are 20 amino acids. So there is a special chart that we can consult called the genetic code. And this is how we would figure it out. So what do you see on this chart? Well, first of all, there are 64 codons. And that makes sense because there are 4 nucleotides, U, C, A, and G. And we have 3 spaces available in a codon. So to figure that out, it's 4, because there's 4 possibilities in each slot. So it's 4 times 4 times 4, which is 64. So there are 64 total combinations possible to create a codon from these 4 nucleotides. So this is it. These are all the possible combinations that we can make. Okay. So let's say I wanted to figure out what our first codon, UGG, which amino acid that coded for. I would look at my chart, and I'm going to start on the left-hand side. So we have the 4 nucleotides, UCAG. So the first one is U. So we're going to be in this portion of the genetic code. The second letter, you're going to go across the top here. So again, we have UCAG. And the second letter was G. So we're going to be in this square, right? The first is U, the second is G, so we're here. For the third letter, which is another G, we're going to look on the right side. So we see again U, C, A, G. So here is U, G, G. and it codes for tryptophan, TRP. And that's what we see here, TRP. Practice one more just so you get the feel of it. So we'll do GGC. So again, we're going to start on the left-hand side and we're going to go down UCAG, G, right? So we're going to be here. Then we're going to go across here and find G. So it's over here. So we're going to be in this box, and then we're going to go across the right, UCAG. Here's this C, because we're doing GGC. So here we go. GGC codes for glycine. And that's what we see here. GLY is the amino acid glycine. Now there's a couple important things to note about the genetic code. Number one, again, there are 64 possible codons. But when you look at this map here, at this chart, you'll notice that multiple codons can code for the same amino acid. For example, UUU and UUC both code for the same amino acid, phenylalanine, PHE. Over here as well, CUU, CUC, CUA, CUG all code for the amino acid LEU, which is leucine. This property has a special term. It's called being redundant. So we have here the genetic code is redundant. More than one codon code for a particular amino acid. So again, we see this all over this chart. So where might this come in handy? Where might this be beneficial for us? Well, if we think about genetic mutations, let's just take this for example. Let's just say, so this of course is a codon, so this is RNA. The DNA, the template DNA for this would be GAA, right? Let's say if this is our codon, so GAA. Let's just say that it was mutated in the DNA so that it is GAC. GAC would create a codon that is CUG here. So although there was a mutation in the DNA, both of these codons both coded for the same amino acid, so that organism would actually not see any effect. That mutation would be a silent mutation. Of course, mutations aren't always silent. If instead the first nitrogenous base was changed, it would give us a different amino acid. So instead of it being a leucine, we would have something else there, and that would change this primary linear structure of our polypeptide. And if the protein polypeptide is altered, it probably won't fold properly. If it doesn't fold properly, it probably won't function as efficiently or at all as it should. So that is the genetic code being redundant. You'll also see that it is not ambiguous, which means that no codon specifies more than one amino acid. In other words, CUU only ever codes for leucine. CGU only ever codes for this one, arginine, right? This codon cannot code for anything else. So we can have multiple codons coding for the same amino acid, but one codon cannot code for multiple amino acids. So the genetic code is redundant and not ambiguous. Last thing that you need to note, and you especially need to note this one, we have start codons and stop codons, and they're just like what they sound like they would be. They are little signals to the ribosomes on when to start making a protein. So the beginning of every single polypeptide always has this start codon, and note this codon, AUG. AUG. AUG codes for a amino acid called methionine, MET, and that is our start signal to start building a polypeptide so we can make our protein. So AUG is the start signal. So imagine if there's any mutation here, we won't have our start signal, so we're not going to make our protein. We have one start signal and we have 3 stop signals. And you don't have to know these. Just know that there are 3 stop signals, 1 start signal, and note what that is, AUG. So back to the idea of the template or non-coding and then our coding strand here. Notice that the template strand is complementary to the coding, of course, and the template is also complementary to the transcript, right? Remember that codons, if you see the word codon, we are talking about RNA. So this is our transcript. Also note that the coding strand and the transcript are very similar. They're almost the same except for the base T and U. So only DNA has T and only RNA has U, but other than that, these two match. So this one is complementary to these two, and these two are very similar. And one last thing with this picture, with DNA, we always have to pay attention to the directionality. So you'll notice that the template strand is always 3' to 5'. That's how it's read. We start at the 3' end and we move toward the 5' end. But if you recall from the previous chapter, when we're building polynucleotides like RNA and DNA, they're built 5' to 3'. This makes sense, right? These two are complementary. So as we're reading this end, the 3' end, we're building this 5' end. As we keep moving from 3 to 5', we're building from 5 to 3'. So the 5' end of the transcript is the front, it's the head, and the 3' end of the RNA transcript is the tail. And we'll see that again. Alright, so that was the basics of the genetic code and codons. Now we're going to get into the actual mechanism of transcription and translation. We're going to begin with transcription. So transcription is the first stage of gene expression. It is the DNA-directed synthesis of RNA. So the whole point of transcription is to read the DNA and create the RNA. We just saw that the RNA is complementary to our DNA template strand. And we know that RNA also utilizes the base pairing rules, the same ones as DNA, except for uracil, which is only in RNA, and thymine, which is only in DNA. And these both pair to adenine A. So we're going to look at 3 separate stages of transcription. We have the initiation stage to get things going. Then we have elongation, which is where we're going to actually be polymerizing, building that RNA polymer. And then the final stage is termination, which is how we're going to end our transcription. And we're going to look at where on the DNA this is occurring and the enzymes that are going to be used. So let's start with this image to get an overview of transcription. So up at the top here, we see our 2 strands of DNA. Again, they're anti-parallel. One of them is 5 to 3' and the other is 3 to 5'. This is going to be important in just a moment. On each strand, there is a region of DNA called the promoter. The promoter is important because this is the signal of where to begin transcription. You cannot start it down here, you cannot start it down here, you can't start it up here. It looks for promoters all along the DNA. And there will be a promoter in front of each gene. So promoters are important to begin transcription. And then the part of the DNA that is going to be read as the template is called the transcription unit. So the first stage is called initiation. Let's take a closer look at initiation. So here we have again our 2 strands of DNA, and these are eukaryotic strands of DNA. So this is a eukaryotic promoter here. Each strand has a promoter. The promoter, again, this is the signal for where your start point is for transcription. The promoter has a special portion of it called the Tata Box. And it's called the Tata Box because it is filled with TA, and then usually a couple of extra A's there. So the top strand here has TA. So there you go. This is the Tata Box. It notes here that the promoters usually extend several dozen nucleotide pairs upstream of the start point. So this whole entire green area is the promoter. You see here you have the start point, which is where transcription is actually going to begin, but this is called, it says upstream, so further toward the beginning part of this strand. Anything later is called downstream. So upstream of the start point, that's where that Tata Box is. Downstream of the start point is our transcription unit. Again, so this here, in this case, when we look at these 2 strands, the bottom strand is going to act as our template. So this is the one that's going to be read. So the top strand here is the non-template coding strand. To begin the process, an assembly of different proteins, which are called transcription factors, are going to bind onto and around the Tata Box region of the promoter. This complex that they form is called the transcription initiation complex. To give you an example, there is a protein called vitamin D receptor, which of course would bind vitamin D, and that can act as a transcription factor. They're not fully understood. We know that they're necessary, but we don't necessarily know all of the proteins that are involved because it's kind of difficult to tease that out. But transcription factors are necessary. They bind onto the Tata Box, and their purpose is to create like a scaffold, something for the main enzyme of this transcription process, which is RNA polymerase II, to bind onto. So we learned about DNA polymerase in the previous chapter, and DNA polymerase read DNA to build more DNA. RNA polymerase reads DNA to build RNA, and this particular one is RNA polymerase II. Once we have created our transcription initiation complex with all of our transcription factors binding onto the Tata Box and the promoter, and the RNA polymerase II is bound on, then we can go into step 2, which is elongation. So this whole process is initiation. Now we're going to go on to elongation. Once the RNA polymerase II enzyme is on there, it is able to polymerize 10 to 20 bases at a time. So it is going to simultaneously untwist the double helix and polymerize. So it has a mechanism where it recognizes the DNA strand, the template strand, which is the dark blue strand, and is adding on the complementary RNA base pairs. Again, this is happening while it unzips the DNA. This is different from DNA polymerase, if you remember. That needed its own enzyme helicase to unzip the DNA. We had topoisomerase also that was helping up in the front, but this is an all-in-one enzyme over here, which is both untwisting and polymerizing. So transcription progress is 40 nucleotides per second in eukaryotes. So it's a little bit slower than DNA polymerase. And we can have a gene that's transcribed simultaneously by several different RNA polymerases. So basically, once this RNA polymerase enzyme jumps onto the promoter and begins to go, another one can jump on behind it and begin too. Now, this is very important. With RNA polymerase and DNA polymerase, it's always important to note the directionality. I always bring this up, so I'm going to point it out once again. We are always building 5' to 3'. We did this when we were building DNA, and we're doing this now that we're building RNA. So again, what this means is the front, the head, this red piece is the RNA transcript that we are building. The front part of it is the 5', and the back part of it is the 3'. So we build 5' to 3'. This means that the DNA template is read opposite. So it's read 3' to 5'. Make sure that you note that. The DNA template is read, the RNA polymerase moves down the DNA template in the 3' to 5' direction while it builds in the 5' to 3' direction. Revisiting this image, we see the same thing. So here is our elongation stage. We have the RNA polymerase that jumped on. The bottom strand is the template. So that's what's being read. It's being read in the 3' to 5' direction. And we have the beginning of the RNA transcript here. It's going to continue as this RNA polymerase moves down the template strand. This transcript elongates, so down here we see it much longer, and it is being built in the 5' to 3' direction. The final step, the third step here, is going to be termination. So once we have elongated the strand and created the transcript of the full gene, how do we stop this process? So to terminate transcription, we have two examples. One is in a bacteria, so this is going to be prokaryotes, and then we'll talk about eukaryotes. Prokaryotes are a little bit less complex, so that RNA polymerase is going to stop transcribing at the end of a part of the DNA called the terminator. And then that mRNA that was created by the RNA polymerase is ready to be translated. So it does not need any other modification. That mRNA can go straight to a ribosome and be created, translated into a protein. In eukaryotes, it's a little bit more complex. So that RNA polymerase 2 transcribes a part of the DNA that results in a polyadenylation signal sequence. Now this word, polyadenylation, poly means many. If you recall the nitrogenous base, A is adenine. So polyadenylation is a bunch of the adenosine nucleic acids in a row. So it will say A. That is called the polyadenylation signal sequence. Once that RNA transcript is complete, it is going to be released 10 to 35 nucleotides past that polyadenylation sequence. And then we're not done yet because this RNA transcript still needs a little bit of modification. So here it is. We're going to talk about what has to be done to that first RNA transcript to get it ready. So this is RNA modification or RNA processing. So first of all, that transcript that was made, that is called a pre-mRNA. A pre-mRNA because, again, we still have to edit it a little bit. And we are still in the nucleus here. So note that. We are still right next to the DNA. We've just made our RNA, and the RNA processing occurs in the nucleus. So what are the modifications? The first thing is that on the 5' end, which is like the head, we're going to put a 5' cap, and this cap is made of guanosine. So we have a G here. So we have our 5' guanosine cap, and on the end, it's almost like putting caps on the two sides of this. On the end, we have to put something called a poly-A tail, which is a bunch of adenosine nucleotides in a row, just a bunch of A's, right? Poly-A. This is not the same as the polyadenylation sequence, which is the termination sequence that we just talked about. So there's mostly A's, we have a U in there as well. So this is a polyadenylation signal. This signals the end of transcription, and then the poly-A tail is the modification that is placed at the 3' end on that pre-mRNA transcript. These particular modifications seem to help facilitate the export of the mRNA into the cytoplasm, because again, remember, we are still in the nucleus and we need to get this mRNA out of the nucleus. So these modifications help export it. They protect the mRNA from hydrolytic enzymes, which are enzymes that would splice it and cut it, and they help ribosomes attach at the 5' end, as we'll see later in translation. And just to get your bearings on all of the components of this pre-mRNA right here, so again, we have the 5' guanosine cap here. We have a 5' UTR, which means untranslated region, which means that this part of the transcript is not going to be made into a protein. Then we have our start codon. If you remember from the beginning of this lecture here, the start codon is AUG, and it's going to code for the amino acid methionine. So this start codon is what's going to signal the ribosome to begin translating. So anything after the start codon is the protein coding part of the transcript. So this contains all of the codons that are going to be used to, here, remember these are our codons. These are what are going to be signaling which amino acids to use to build our protein. After the protein coding segment, you have the stop codon. Remember, there are 3 stop codons, 1, 2, 3. And after the stop codon, we have the 3' UTR, which is the untranslated region. So again, this part's not being made into a protein. This is that polyadenylation signal, which is what signaled the stop of transcription. And after the polyadenylation signal, we place the poly-A tail as a little end cap here. But we're still not done. There's one more important step that we have to go through, which is called splicing, RNA splicing. So most of the eukaryotic genes and their RNA transcripts have long, non-coding segments of nucleotides that are in between coding regions. So here is the pre-mRNA that we've just prepared. We had our RNA transcript that was created right from the DNA. We stuck our 5' cap on. We stuck our poly-A tail on. But you'll see here that there are these darker red and lighter pink colored regions. Those represent parts of the transcript that are coding and non-coding, meaning parts that make proteins and don't make proteins. So the point of this is to create a protein, right? So we want to splice out the parts that are not going to code for a protein. So those 2 regions are called exons and introns. Exons are the protein coding regions. So that's these dark red colors here. So there's 1, 2, 3 exons in this transcript. The introns are the non-coding, so we're going to cut those out. And the way that I remember that is that introns interrupt. So we don't want any interruption, so we're going to splice out. Splice just means that we're cutting it out, and then we're going to glue the other pieces on each side together. So we're going to splice out this intron and splice out that intron. And then we are left with just our exons, which is our coding regions. So we have all the dark red here. We still have our 5' cap. We still have our poly A tail. We still have our 5' UTR and our 3' UTR. And of course, in here is also going to be our start codon and our stop codon. And now this is an mRNA, not a pre-mRNA. We have put on our modifications and we've spliced it so it is an mRNA. This mRNA can now exit the nucleus and move toward ribosomes for translation. So now we're going to move on to the second major part of gene expression, which is translation. So translation is the RNA-directed synthesis of a polypeptide. So in other words, the genetic information is flowing from that mRNA that we've just prepared to a protein. Now we're going to cover a couple of extra types of RNA. So we've just went through mRNA, and now we're going to learn about tRNA and rRNA, which we actually talked about a little bit in chapter 6. But all three of those are involved in translation, all three of those RNAs. And we can actually see all three types of RNA in this image right here. This red ribbon is of course the mRNA. Here's our 5' end, here's our 3' end. This two-segmented brown thing is called the ribosome. We're going to get into the ribosome in a minute, but the ribosome, if you recall from chapter 6, is made up of rRNA. R means ribosomal, so ribosomal RNA makes up the ribosome. And then the third type of RNA is the tRNA. The tRNA is what's carrying each of the amino acids, which are shown here as purple shapes. So all these purple shapes are representing the 20 different amino acids, and the tRNAs, it's their job to carry one at a time and bring it to the ribosome. So those are the three types of RNA. Let's go deeper into tRNA. So again, tRNA stands for transfer RNA because its job is to transfer an amino acid onto the protein that we're trying to create. So take a look at the tRNA here. Notice that you see it drawn a couple of ways here. It's kind of drawn in this cross-shaped T, but notice that it is one strand. Remember, DNA is a double-stranded, but RNA is single-stranded. So this is one continuous strand, which does double back on itself, and there is complementary base pairing. So that absolutely still happens. And that affects its 3D structure, which is what we see here. So it kind of, I don't know what shape this is, but it's that shaped. That is a tRNA. So the tRNA is carrying the amino acid at the 3' end, which is at the top over here. And down at the bottom of the tRNA is a very important part called the anticodon. Now you know where the codons are. Let's go back a slide. The codons are all located on this red ribbon, which is the mRNA, right? Here are 3 codons, UGG, UUU, GGC. Those are 3 different codons. The tRNAs, which here are represented like this, the tRNAs at the bottom have the anticodon. The anticodon carries the complementary base pairs to the codons. That's how they match. So this is very specific in which tRNA, which has its own anticodon, which amino acid that tRNA is carrying. So as we see here, this anticodon is CCG. It's going to base pair with the mRNA's GGC, right? C is binding with G, C is binding with G, G is binding with C. Anticodon matches the codon. Now, if this tRNA was carrying the wrong amino acid, or just any amino acid, then it wouldn't put on the proper one, right? So these are specifically matched. This CCG anticodon is always going to be carrying this amino acid glycine, for example. So just to see it one more time, here is our single strand of RNA, tRNA, the 3' end. Remember, we always add things onto the 3' end. The 3' has that hydroxyl group, which I call like a sticky OH, a sticky hydroxyl. So when we're building RNA or DNA, we always build onto the 3' end. So it's the same here. The 3' end is going to grab onto the amino acid. The 5' end is over here. And down here at the bottom is where the anticodon is. It's 3 base pairs. Here it's AAG. And here's the other representation, the cartoon representation of the tRNA. Again, AAG, 3' to 5' here. So you see they mentioned something called Wobble. Wobble allows for flexible pairing of the 3rd base of the codon. So we're talking about G here. So the ideal codon to this anticodon would be UUC, right? UUC. So with Wobble, this tRNA with this anticodon might be able to bind to UUG instead. The 3rd base pair might not be the correct one. This allows the tRNA to bind to multiple codons. You might think that this is bad and this could result in binding of the wrong amino acid there, but if you look back at the genetic code, you'll notice that very often, let's just look at leucine here, the first 2 base pairs are the same, CU, CU. The 3rd one is different. In fact, any of the 4 nucleotides could bind in this 3rd position and would still give you leucine. We see that for a lot of them. We have threonine, alanine, valine, right? The 3rd position is different, but the first 2, first and second, are the same. So Wobble wouldn't really affect this at all. So that was tRNA. Let's look at the ribosomes, which carry the rRNA. rRNA just means ribosomal RNA. So the ribosomes are what facilitate the coupling of tRNA anticodons with the mRNA codons. Ribosomes contain 2 subunits. They have a larger subunit and a smaller subunit. Within this larger subunit on top, you'll notice that there are 3 different binding sites that are important to note and understand. So we have EPA. E stands for exit, P stands for peptidyl RNA, peptidyl tRNA, and A stands for amino acyl tRNA. So those are big words, but you'll see what they are in a second. It's not so bad. What you're going to see is that when we are going to translate a protein by reading an mRNA, the mRNA is going to be entering from this way and going across. And remember, we put on a 5' cap. So remember that the cap is the head goes in first. So the 5' end is going to go from A to P to E. And of course, E is exit, right? So these sites correspond to the codons and the tRNA's ability to bind properly with their anticodon and getting that amino acid onto our ever-growing strand. We're gonna see when we talk about that stage of translation. I'll walk through it even more. All right, now you know all of the components that are required for translation. We're gonna walk through the actual steps. So just like with transcription, translation has 3 stages: initiation, elongation, and termination. They're even called the same. So be very diligent in how you take notes on this, because I just find that students often confuse the initiation of transcription with translation, the elongation of the two of them, specifically the initiation. What I would suggest is creating a table, something just like this, so that you can properly organize this. Organization is very important for bio, with being able to recall this information properly, because often there's a lot of similarities, and the questions, when you get to a little bit higher level of questions that aren't just plain, can you remember what this means, is often comparing and contrasting between similar things. So transcription, translation, then we have our initiation, our elongation, and our termination. And what you want to do is list all of the components that are on your slides, what they do. Just put all the details into a table just like this. So we're going to walk through the 3 stages of translation, and note that all 3 stages are going to require protein factors that are going to aid in the translation process. First up, we have initiation of translation. So remember, the ribosome that we just learned about had 2 subunits. When a translation first starts, those 2 subunits are not together. They're separate. So we start with the smaller subunit, the small ribosomal subunit here. What happens is the mRNA, which contains that 5' cap, that 5' cap helps it to associate properly and align itself with this part of the ribosome. So it's going to feed right through until the ribosome finds the start codon, right? The start codon, remember, write this down. It's over here for you. AUG. AUG always codes for the amino acid MET, which is methionine. So this is methionine. So our start codon, which is on the mRNA, is recognized by the proper tRNA, because the tRNA is carrying the anticodon. So the red is the codon here, AUG. Here's our anticodon, UAC, right? Those are the proper complementary base pairs there. The tRNA associates, we have methionine. Now, with a little bit of energy, we have a GTP, which is just like ATP. The large ribosomal subunit is able to click on to the small ribosomal subunit, and now we would be ready to continue the translation process. That large ribosomal subunit was brought in using proteins called initiation factors. So just note that in your notes. Initiation factors are going to bring in this large subunit so that it can connect onto the small subunit and begin this is called the Ribosome Association, and this is the initiation for translation. So now what we have to do is we're going to read our mRNA and build that polypeptide. So this is the elongation phase. This is where we're actually building our protein. So let's start here in this cycle. So this is a little bit further on, but it's basically where we just were. We created our Ribosome Association with a tRNA in the P site here. And that tRNA was carrying one amino acid methionine. That's just like here. So the next thing is that to the right in the A site carries a new codon, right? Those 3 red nucleotides right there. That's a codon that will be read by a tRNA. So the next tRNA, which carries the anticodon matching the codon in the A site, is going to bind, and that tRNA is carrying its proper amino acid. So that's here. So here's the anticodon and codon associated. Now, the tRNA that is in the P site is what is carrying the polypeptide chain that we're building. So you see this one already has a couple on it. So how do we get these connected? The tRNA from the P site passes this whole chain onto the tRNA in the A site. So this purple circle is going to be connected onto this purple diamond over here. That leaves us at this image, right? So now our tRNA in our P site no longer has any amino acids associated. They were all just added right onto here. So now what happens is the ribosome clicks over. So these aren't moving. The ribosome moves. The ribosome is going to click one codon over. So what happens is this E site becomes filled by this tRNA here, and this, which was in the A site, is now in the P site, and the A site is once again open so that the new tRNA can come bind. So here's a representation of that. Here's the tRNA that was this one, and it is no longer carrying anything. It's in the E site. Remember, E stands for exit. So this tRNA is going to exit. The P site, once again, is carrying the tRNA with the polypeptide chain. You see that it's longer now. We started off with 1, 2, 3, 4 amino acids. Now it has 5. And here's the A site open once again. So this codon is now going to be read, and the proper amino tRNA carrying the amino acid is going to bind. This is going to happen all the way down, and again, translation proceeds along the mRNA 5' first, 5' cap to 3'. And it's going to happen all the way down through the coding region, all across those codons. And then finally, we are going to terminate our translation. So this is the third stage, termination. Termination occurs when the ribosome finds that stop codon. So remember, we have 3 stop codons. They are listed here for you, UAG, UAA, and UGA. When the ribosome comes across a stop codon, in the A site, the A site is going to accept a protein which is called a release factor. So that's this yellow protein. The release factor binds into the A site, and this reaction releases that polypeptide that we just created. This is our linear chain of amino acids, and then this whole translation assembly comes apart. Now that free polypeptide is going to need to be folded. It can simultaneously coil. It can be sent into chaperonins to be folded into its proper 3D shape. That way that polypeptide becomes a functioning protein. Sometimes, often, proteins also would need to go through some sort of post-translational modification. This can be anything from adding sugars or lipids onto it or cutting pieces off. Now, there's an amplification process in our cell. It takes a lot to open the DNA, to read it, to transcribe it into an mRNA, and to get that mRNA over to the cytosol. So with that one mRNA transcript, we can actually read it with many ribosomes and make many of those polypeptides, those proteins. So this is called polyribosomes. So they enable a cell to make many copies of a polypeptide very quickly. You can see on this chain, this mRNA right here has, I don't know, maybe 15 or 20 ribosomes associated with it. So they all jump onto the 5' end, and they move down, and as they do that, they're translating and building their protein. You may remember from chapter 6, we talked about ribosomes when we talked about the rough ER. Remember, there's smooth ER and rough ER. And the rough ER is called rough because it is studded with ribosomes. So we have two populations of ribosomes in the cells. They're either bound to the ER, bound ribosomes, or there are ones that are just free floating in the cytosol. The free ribosomes are mostly involved with synthesizing proteins that function within the cytosol, whereas the ones that are bound to the ER make up proteins of the endomembrane system and proteins that are going to be secreted, which means sent out of the cell. And ribosomes are identical whether they're free or bound, so they can switch from being free to bound. For the last part of this chapter, we're just going to go over mutations. So mutations are changes in the genetic material of the cell. So essentially, we are changing the sequence of ATCG, right? So mutations of one or more nucleotides can affect protein structure and function. This, of course, all depends on where in the DNA structure that mutation takes place. Mutations can result from spontaneous changes that occur during DNA replication or recombination during meiosis 1, or while there's a little bit of repair that's going on. If there's any errors in any of these molecular processes, we could end up with a spontaneous mutation. Or there are agents, physical or chemical agents, that can cause mutations. An example of a physical agent would be UV light. And then if you ever work in a lab, there are some chemicals that are around, and they'll be labeled, and you'll know that they're DNA mutagens. And a mutagen is just something that can alter the structure of your DNA. So let's look at point mutations first. A point mutation is a change in just one base pair of a gene. Let's look at this example that we have here. We're looking at the protein hemoglobin, which is involved with carrying oxygen through your blood. So we have wild-type hemoglobin, which is the good hemoglobin, and we have the sickle cell hemoglobin. If you recall, we did touch on this when we were learning about proteins. Essentially, if the hemoglobin protein has a mutation in it, it causes it to fold improperly. It kind of makes it sticky, and that cell sickles. And then if the cell is sickled, then it can't properly carry oxygen, which is of course not good. So we have here the wild-type hemoglobin, DNA, CTT, and GAA. Our template here is the top strand, the 3' to 5' strand. And from this strand, we create an mRNA that is GAA. GAA codes for the amino acid glutamine, GLU. So this is a normal hemoglobin. Here is our point mutation with one base pair of the gene different than what it should be. Instead of a T and an A, we have an A and a T. So this, again, the top is the template strand. We create from this GUA instead of GAA, and GUA codes for the amino acid valine. So this sickle cell hemoglobin is mutated. What we just saw was an example of a nucleotide pair substitution. In a nucleotide pair substitution, this replaces one nucleotide and its partner with another pair of nucleotides. So that was this pair was replaced by this pair. So that's a nucleotide pair substitution. So these are the outcomes that we can have from mutations. We can have a silent mutation, which has no effect on the amino acid that's produced by our codons because of, if you remember I showed you earlier, the redundancy of the genetic code. Let's look at the genetic code. Remember, the genetic code is redundant and not ambiguous. This means redundant means that more than one codon can specify for a particular amino acid. So for example, if the codon was supposed to be CCC and somehow it got changed to CCA, this is not a big deal, even though this is a point mutation right here, because they both still code for the same amino acid. So that point mutation would be silent. You can have missense mutations, which still code for an amino acid, but for the wrong amino acid. So looking back at our genetic code, if it was supposed to be AUA and instead there was a point mutation and it was AUG, AUA would have coded for isoleucine up here. AUG codes for methionine, which is the start codon, and that will mess up our protein. So that is an example of a missense mutation. And we also have nonsense mutations, in which there's a change in the amino acid codon into a stop codon, which will lead to a non-functional protein. So looking back at our genetic code, if it's supposed to be, let's say it should be CAG, and instead it's turned into UAG, that's a stop codon. So anything that gets changed into either UAG, UAA, or UGA is a nonsense mutation. This will lead to a non-functional protein. We can also have insertions and deletions, which are especially bad, and we'll see why in just a second. An insertion is an addition of nucleotide pairs, and a deletion is removing of nucleotide pairs from a gene. These are much worse than the substitutions that we were just looking at. The three that we just saw, the silent mutation, missense mutation, nonsense mutation, those were just substitutions, right? One pair of nucleotides was switched out. But here, we are inserting or removing whole pairs of nucleotides. So what this does is it causes a frameshift mutation. So it's going to alter what's called the reading frame. So let's look at that. Here's a comparison between a nucleotide pair substitution, right, where we're just substituting a pair of nucleotides. So we're creating the wrong codon, versus a nucleotide pair insertion or deletion. So up here is the wild type gene. This is what the DNA template strand and coding strand and mRNA should look like in a healthy, well-expressed gene. So here are the 3 examples of nucleotide pair substitutions: the silent mutation, the missense, and the nonsense. We see that in the silent mutation here, this nucleotide should be a G, and instead it's an A, right? So here's the GC, and instead we have AT. This changed our codon from GGC to GGU, but we still coded for the amino acid glycine, so that's why this is silent. There's no effect on the amino acid sequence here. In the next example, we're looking at a missense. So this pair should be a CG, and instead of it, it's a TA. So this changed this code here from GGC, the codon, to AGC. And this did change the amino acid. It should have been a glycine, and instead we get a serine. So we're still making a protein, and we have the wrong amino acid. So this is a missense, and this is going to affect how it folds and probably how it functions. Here we have a nonsense mutation. So this is supposed to be a T and an A, and instead we have A and T. So this codon should have been AAG, coding for this amino acid, and instead we have UAG, which is a stop codon. So this is a nonsense mutation. This won't result in any protein. Over here on the right, this is our insertion and deletion examples here. So for the first example, we see here that there's an insertion of a base pair. It should read TAC-TT, and instead it reads TAC-ATT. So this pair has been inserted. Because of that, we have an extra nucleotide in the codon, in the mRNA here. And what that did was it shifted everything. So it should say UAG, AAG. But instead, since we have to read every 3, inserting this U in here shifted the frame, the reading frame, so that instead of this being the first, second, and third, this is now the first, second, and third. So this created a new codon. In fact, it created a stop codon. So this frame shift caused an immediate nonsense mutation here. In the next example, we are missing a nucleotide base pair. So it should read, let's go here, it should read AAACC, and instead it reads AACC. So we lost out on an AT base pair here. Because of that, we are missing out on a nucleotide here. Once again, this shifts all of our codons. So the third codon should read UUU, and instead it reads UUG, because G would be the next one. So since this U was taken away, all of this shifts. So this G becomes part of this codon. And because of that, UUG codes for something else, leucine. So this one got messed up. Usually with frame shifts, all of the, or a lot of the amino acids get messed up downstream of the insertion or deletion because of that frame shift. So this was a missense mutation because it still did code for amino acids, but it's probably just going to make a protein that is not at all functional here. And then finally down here, we don't have a frame shift, but one amino acid is missing. If you look up here, we're looking at the first couple of nucleotides. We have TAC, TTC. Here we have TAC, and TTC was removed. So it wasn't just one nucleotide that's missing, 3 are missing, and that corresponded to an entire full codon. So there's no frame shift because all of the downstream codons are still read properly. We're still creating phenylalanine and glycine, right? Here, PHE and GLY, but we're missing one amino acid. In some ways, this might be better than if we had just missed one nucleotide, because all of these are still okay. All of the amino acids that would be built here downstream are still okay. Okay, and that wraps up Chapter 17, Gene Expression. You did it. You made it through Bio 101. So for this chapter, I really do recommend making the table that I suggested for transcription and translation. Students often confuse those details, so you want to get them really well organized for yourself. And good luck.